Good morning, everyone. Welcome to the August webinar. My name is Rebecca Bailey, and I will be the moderator for today's webinar. We have one housekeeping item. You can enter questions for the speaker today at any time during the webinar by entering the questions into the box on your GoToWebinar side panel. Our speaker today will be taking the questions at the end of the webinar. Today, I am very pleased to introduce our speaker, Dr. Sheena Sharma. Sheena is a research investigator within the PBPK, Clinical Pharmacology and Pharmacometrics team at Bristol Myers Squibb. Prior to this, Sheena completed her PhD in Dr. Prasad's lab at the Department of Pharmaceutical Sciences at Washington State University in Spokane. She holds a bachelor's degree in pharmacy from the Delhi Institute of Pharmaceutical Sciences and Research, and a master's degree in pharmaceutical analysis from the National Institute of Pharmaceutical Education and Research in India. Through her training in model-informed drug development, Sheena has nurtured a profound interest in predicting the impact of physiological and drug-related factors on drug exposure and response. She's the author of several research articles in diverse journals, such as the Journal of Clinical Pharmacology and Molecular Pharmaceutics. And Sheena joins us today as she was recommended as a interesting and great speaker from our early clinical researchers initiative. And so with that, I am really pleased to welcome Dr. Sheena Sharma. Thank you.
Thank you, Rebecca, for the nice introduction, and also thanks to Rosa Webinars for this opportunity to present my work. I would also like to thank everyone for joining this webinar, and today I will be talking about proteomics-informed PBPK modeling to predict systemic and tissue drug concentrations in rats. I would like to share a disclaimer that the presented work was conducted in Dr. Bhagwat Prasad's lab as part of my PhD dissertation, and the work was funded by the College of Pharmacy and Pharmaceutical Sciences at Washington State University, Spokane. If you haven't been to Spokane yet, it's a very nice small town, about 5 hours drive from Seattle, and it is also a birthplace of Father's Day. Also, thanks to all the co-authors who have contributed to the publication in molecular pharmaceutics about the work that I'm going to present in this presentation today. Just a brief outline of the presentation today. I'll be talking about the importance of tissue drug concentration, quantitative proteomics, and the integration of proteomics data in PBPK modeling. I'll be also sharing a case example of digoxin where we predicted the tissue drug concentrations. And finally, I will be concluding the talk. So let's start with the problem. The problem is high rate of failure in drug development. So over 90% of drugs fail in the market. Only 1 in 10 drugs is successful in the clinic. Drug development takes over 10 years and more than $2 billion, and most of the money being spent in both discovery as well as clinical drug development phases. So let's review some of the reasons of failing drugs in the market. The pie chart here presents data which was analyzed between 2013 and 2015. Among many reasons, the most important ones include the lack of efficacy and safety concerns. So over 70% of the drugs fail in the market due to either lack of efficacy or safety. This is partially because measuring drug concentrations in target tissues is often not feasible where efficacy or toxicity occurs. And this also leads to low confidence in target occupancy profiles, which is a key information for efficacy assessment as well. One of the techniques to measure tissue concentration of drugs include positron emission tomography or PET imaging. As shown in the figures here, the PET imaging can be used to calculate the standard uptake values or the SEUV index, and it can be done throughout all the tissues. However, this technique is costly and also logistically challenging. This highlights the need to predict tissue concentrations of drugs, especially when transporters are involved. So, as an alternative to measure tissue concentrations, it can be predicted using in vitro assay, quantitative proteomics, and PBPK modeling. And to do so, quantitative targeted proteomics can be employed. Proteomics information is needed to quantify the protein abundance in vitro models, including transporter expressing cell lines, the membrane vesicles, and also the protein abundance can be measured in the tissues. This information is needed to calculate the relative expression factor, which is a physiologic scalar to normalize or correct the protein abundance between the two different systems, in vitro and in vivo. And that's why it's a ratio of in vivo protein abundance to in vitro protein abundance. This ref scalar is used to correct the transporter based drug clearance, and this further can be integrated in the PBPK modeling. Now, in order to demonstrate the importance of tissue drug concentrations in driving the pharmacodynamic or response or the PD response, I have a case example of rosuvastatin where Rose and colleagues performed the PBPK PD analysis. The model was built and verified at different doses. I am presenting one of the key figures here. So rosuvastatin is a cholesterol lowering drug and the liver is its main site of efficacy. Within the liver tissue, it's transported by different drug transporters, including the OATP1B1 transporter, which is a basolateral uptake transporter. Here in these graphs, I have the concentration time profile curves. So the first graph has the simulated rosuvastatin plasma concentration in different OATP1B1 genotypes. So the solid red line represent the reference type or the genotype with the extensive transport. The dotted green line represent heterozygous condition intermediate transport, and the blue dotted line represent the homozygous deficient or the poor transporter activity. So basically, with the increased or the extensive transporter and basically more clearance, we have less rosuvastatin plasma concentration. Now, if we look at the second graph, we have the liver unbound intracellular water concentration of rosuvastatin, and in this case, there is basically no effect of the genotype on the predicted liver intracellular concentrations. And the model also assessed the effect of the OATP1B1 transporter genotypes on the drug response, which is the plasma mevalonic acid concentration. And here, the PD was assessed with both plasma as the driving concentration for the pharmacodynamic response, as well as the liver as the driving concentration for pharmacodynamic response. So as shown here, the the model when the liver concentration was used as the driving concentration for pharmacodynamic response is more consistent with the clinical data because the clinical data, the response basically is not affected by the genetic polymorphism. And here in the lower two graphs, we can see that when we are using the plasma concentration as the driving concentration for the pharmacodynamic response, the genetic polymorphism affects the mevalonic acid concentrations as well in the plasma. However, in clinic, there is actually minimal effect of this genetic polymorphism on the mevalonic acid concentration or the cholesterol synthesis, which is a more mechanistic understanding of the statin action. So this basically shows that this using the liver concentration as the driving concentration for PD is more mechanistic to explain the statin action. A sensitivity analysis of the influence of total uptake intrinsic clearance or the CLINT by OATP1B1 transporter was also performed. In different graphs here, there is total clearance intrinsic by OATP1B transporter as the x-axis, and on the y-axis in the first graph there is plasma AUC, then second has the liver unbound intracellular concentration or the AUC, then the third represent the muscle AUC, and finally we have the reduction in the mevalonic acid AUC. The last graph represent the elasticity index, which is a measure of the relative change in selected variable to the relative change in the clearance intrinsic. It's basically a normalizing the scale for comparison of the sensitivity of multiple model output parameters. So the key message here is that here, the plasma drug concentration serves as a surrogate of toxicity, which is the muscle drug concentrations. So basically, by increasing the total intrinsic clearance of the transporter, uptake transporter OATP1B1, there is a decrease in the plasma AUC as well as decrease in the muscle AUC. However, by increasing the total clearance intrinsic, the liver unbound intracellular AUC increases. Similarly, the mevalonic acid AUC, the reduction in the mevalonic acid AUC is in parallel with the liver unbound concentrations. So this represents that the liver concentrations serve as a surrogate of the PD response or the mevalonic acid concentrations. However, the plasma drug concentration is a surrogate of toxicity or the muscle drug concentrations. So let's get back to the problem of high drug failure in drug development. And in this direction, Morgan et al. Published this paper where they shared the learnings that can be applied to improve compound survival in an analysis performed on the data analysis from phase 2 decisions for 44 programs at Pfizer, and they proposed these 3-pillar risk management metrics for mechanism testing. And one of the key pillars, the first pillar, involves the exposure at the target site of action, so basically highlighting the importance of tissue drug concentrations. The other two pillars include binding to the pharmacological target and expression of pharmacology. So a combination of these 3 pillars builds this metrics, which is a combination of the exposure confidence and the confidence in pharmacology. So basically, with having confidence in these 3 pillars, we can actually maximize the confidence in drug translation. So this highlights that in order to navigate to the drug development stages, having the confidence in target site of action, so one of the pillars, is important. Since we are talking about navigating in drug development phases, it's important to review what data do we mostly have in the early phases of drug development. So mostly we deal with the in vitro, the data coming from in vitro systems, preclinical species, for example, mice, rat, dog, and monkey. And based on this data, we perform the allometric scaling from preclinical species to extrapolate to humans. However, in most cases, it's challenging to perform the allometric scaling merely based on body surface area or body weight, especially when the drug is majorly metabolized and transported. And this is because the differences in the drug-metabolizing enzymes and transporter are abbreviated as DMET throughout my presentation here. So the differences in the DMET protein abundance and orthology or the sequence similarity lead to the interspecies variability in drug systemic as well as local pharmacokinetics and therefore pharmacodynamic the response. So therefore it's important to understand these differences in the drug metabolizing enzyme and transporter protein abundance in order to adequately translate the findings from preclinical species to humans. Here, just as a recap, I want to illustrate the vast majority of drug-metabolizing enzymes and transporter proteins present in the human liver, kidney, and intestine, which are the major organs of drug elimination. So as shown here, a number of different enzymes and transporters play a role in drug disposition and also absorption in different segments of intestine. And in while we are thinking of extrapolating data from preclinical species to humans, it's important to pay adequate attention to the differences in these DMET protein abundance, which can basically affect the drug pharmacokinetics and therefore pharmacodynamic response. So I want to here illustrate the interspecies differences in the DMET abundance. So just to highlight some of the proteins which are different between rats and humans. So for example, the PGP transporter, it's an efflux transporter. Also, it's one of the site where it's majorly expressed is the blood-brain barrier, and it's higher in the rats as compared to humans. So while using rat model for testing a drug uh, efficacy, it can show it can lead to the lower brain concentrations in case of the rats as compared to the humans. Likewise, the UGT1A10 enzyme, it's higher in the enterocytes, human enterocytes. However, the same enzyme UGT1A10 is higher in the rat liver as compared to the human liver. So some of these differences in the observed efficacy and safety of drugs between humans and rats can be partially explained by physiological differences in the DMET protein abundance between these two species. If you're wondering if there are any real life examples where these interspecies differences led to the failure of drug candidates in certain phases of development, then the answer is certainly yes. For example, this compound by J&J and the SGX 523, they failed as clinical candidates because of nephrotoxicity in humans. And this is likely because of the higher aldehyde oxidase abundance in humans compared to rodents, which led to the accumulation of the AOX-mediated insoluble metabolites in human kidneys, which was not caught earlier while the drug was being tested in rodents. Rodents are also sometimes not reliable in translating hepatotoxicity to humans. For example, rats fail to predict acetaminophen-induced liver injury, explained partially by the interspecies differences in CYP2E1 mediated formation of the hepatotoxic metabolite. Similarly, the breast cancer resistance protein or PCRP abundance is 50-fold higher in the rat kidney compared to the human kidneys, leading to the differences in the toxicity as well. So the question here is, how can animal data be translated to human considering the interspecies differences in DMET protein abundance? And one of the reliable alternative is using physiologically based pharmacokinetic modeling to predict drug absorption and disposition, including tissue drug concentrations. However, these PBPK models require comprehensive data on drug and physiology specific parameters, including the abundance of DMET proteins. And so to do so, the present study aimed to develop a repository of rat tissue quantitative proteomics data, which can then be used to predict systemic and tissue drug concentrations in rats prior to human studies by performing proteomics-informed PBPK modeling. This is the first comprehensive study reporting the quantitative proteomics abundance data in rat liver and intestine, which is useful in delineating the pharmacokinetics and toxicity profiles in rats for better extrapolation to humans. It's also important to highlight that even the regulatory agencies, they have been encouraging the use of PBPK modeling in different applications, including biopharmaceutics and spatial populations. So the FDA, EMA, as well as PMDA, there are also the Canadian the regulatory agencies. They all are very much in favor of using these PBPK modeling tools as well as the QSP modeling to support the drug labeling as well as the trial design, protocol design submissions. Recently, the FDA Modernization Act was also passed by the Senate, And this bill authorizes the use of certain alternatives to animal testing, including the cell-based assay and computer modelings, for example, including the PBPK modeling to obtain exemption from the FDA to investigate the safety and effectiveness of a drug. So again, this calls for action, and the use of PBPK modeling and the QSP modeling is definitely something which is encouraged by the regulatory agencies to support the drug efficacy as well as the safety. Now I would like to share the workflow or the methodology for performing quantitative DMET proteomics. So to start, we basically performed the protein isolation and trypsin digestion on the Sprague-Dowley rat tissues, including liver and the regional intestinal segments, the duodenum, jejunum, and the ileum and colon segments. And then once the samples were performed, the proteomics, the both untargeted proteomics and targeted proteomics, they were performed. So as the name suggests, untargeted proteomics refers to the quantification of all the proteins that are present in a given sample, and it uses a total protein approach method where the protein abundance is calculated as a ratio of the MS response of that particular protein to the sum of the MS1 spectral intensities of all peptides matching sequence of protein X. And in the targeted proteomics, it basically requires a synthetic unlabeled peptides as calibrators, and it can only quantify the proteins of interest. So we use these two techniques to one untargeted proteomics was used to get the comprehensive list of all the transporters and the enzymes, drug transporters and drug metabolizing enzymes present in the rat tissue samples. And then selectively, some of the proteins were also verified. Their protein abundance was also verified using the targeted proteomics approach. So once having we had this information, then we used this data for some of the proteins to showcase the utility of the protein abundance data in PBPK modeling to predict systemic and tissue concentrations of digoxin. Apologies for a busy slide here, but what I want to show is the results from the untargeted or global proteomics map for the DMET protein abundance across the rat tissues. So here I'm showing the results in the liver and the jejunal compartments. However, the analysis was also performed on the duodenum, ileum, and colon segments of rat intestines as well. So on the x-axis is the drug metabolizing enzymes, a different majority of the drug metabolizing enzymes and also transporters present in the rat tissues. On the y-axis is the mean abundance of these DMET proteins in the picomole per gram tissue unit. So overall, the global proteomics based total protein approach was able to quantify 66 DMET proteins in the liver and 37 DMET proteins in the intestinal segments of Sprague-Dowley rats. Inconsistent with the human observations proteomics data, CYP and UGT enzymes were also mainly detected in the rat liver, just like we observe in the human liver. PGP abundance was also observed to be higher in the intestine as compared to that in the liver, which is also consistent with the human observations. BCRP was most abundant in the intestinal segments, and some of the transporters like OATP 1A1 and the MRP26, they were predominantly detected in the liver. So these observations were pretty consistent with what we observe in the human tissues as well. For some selected proteins, targeted proteomics was also performed to verify the findings in the global proteomics experiment. So similarly, there is the list of the DMET proteins on the x-axis and the mean abundance on the y-axis. The abundance of most of the DMET proteins determined by the global or untargeted as well as the targeted proteomics analysis was within threefold. So these DMETs includes the CYP and the CYP enzymes, UGTs, as well as the transporter like MRP2. However, some proteins like CYP 2C13, the BCRP transporter, NTCP, and PGP transporter, they showed more than threefold difference between these two proteomics methods, that is the targeted and the untargeted or global proteomics methods. And the proteins that showed larger differences in the protein quantification by the global and targeted data were mostly the low abundant proteins. The second goal of this analysis, after quantifying the protein abundance, DMET protein abundance in different rat tissues, was to integrate the DMET protein abundance data in PBPK modeling. And so for the same, we use digoxin as a model drug to show the utility of proteomics data in PBPK modeling. Digoxin is used for heart failure and arrhythmias, and it's in aerotherapeutic index drugs. It's a low solubility, low permeability compound with bioavailability ranging between 50 to 90%. It's a substrate of PGP efflux transporter. So before going into the result of the proteomics informed PBPK modeling data, I have a few slides about the background of building PBPK models. So I can first show the workflow of building a PBPK model. So usually we start with building an intravenous disposition model where both drug dependent and physiology related parameters are incorporated in the model, and the model is verified against the observed IV data. In cases where the model is not able to capture the observed data, then the parameters, including the clearance and the distribution parameters, they are optimized, and the IV disposition model is extended to build the oral absorption model, where the same clearance and distribution parameters are used in addition to the absorption related parameters, for example, solubility, permeability, dissolution, or the intestinal metabolism and transport as well. And in this case, the model is verified against the observed oral data. So in order to review the approaches for building a PBPK model, there are mainly three approaches: bottom up, top down, and middle out. In a sense, PBPK modeling is bottom up in nature. It's the mechanistic, so it can accounts for the non-linearity, and it starts from the intrinsic clearance to calculating the organ clearance. And one of the downside is sometimes the in vitro data is not able to recapitulate the observed in vivo data, so the bottom up can lead to less accurate predictions. In the top-down approach, we start from the organ clearance to calculate the intrinsic clearance, and it can account only for the linearity, but it supports the clinical trial decision and it's suitable for population PK analysis. So the best of both worlds is by the middle out approach, where we use both in vitro and in vivo data. It's semi-mechanistic in nature and yet it's clinically relevant. Among the tissue models, there is an option to basically decide whether a tissue is perfusion or permeability rate limited model. And in general, the perfusion rate limited model, tissue model, is when the permeability of a drug is permeability surface area product is way higher than the blood flow, organ blood flow. And the permeability rate limited model means that the permeability surface area product is much lower than the organ blood. So whichever of these parameter, the permeability surface area product and the blood flow are less, determines whether they are rate limiting or not. And in a sense, they are different because in the perfusion rate limited model, the transport between the intracellular and the interstitial space is not accounted. One of the common misconceptions is that the drugs that are metabolized are perfusion or blood flow rate limited, but if the permeability of drug is low, meaning they are hydrophilic or transporter role it may be negligible, then also we can use the permeability rate limited model. The validation criteria for PBPK modeling depends on the purpose of the study, but mainly bioequivalence criteria or twofold criteria are two of the most used validation criteria for modeling exercise. Bioequivalence criteria is suggests that the simulated to the observed PK endpoints AUC or Cmax lies within the 0.85 to 1.254. It's a bit more stringent but can be used for dosing regimen and waiver of clinical trials. On the other side, twofold criteria is a bit on the looser side, but it can be used for the trial design or to assess the sampling time point collections. Review the results of the proteomics informed PBPK modeling of digoxin. I don't want to bother too much with the equations here, but just want to highlight the places where the DMET protein abundance information was used in the model and how it was incorporated. So in the PKSIM, we have an option to provide the relative abundance data. Protein abundance in intestine or kidney was calculated as a ratio of the abundance lever. So for example, the relative PGP abundance was calculated with respect to the liver in the kidney, jejunum, ileum, and colon tissues. And this information was provided in the model. So first, the IV disposition model was built and verified against the observed data in rats. And then the same model was used to predict the drug concentrations, drug exposure following an intraperitoneal administration and the per oral administration of the digoxin. So in all these simulations, the observed and the simulated exposure was within twofold, which was our validation criteria. And in PKSIM, there is an option to use the permeability rate limited tissues. So for digoxin, the already the PGP transporter is involved and the permeability is less, so we use the permeability rate limited model in using the PKSIM software. Then next, after building and verifying the model for IV disposition and oral absorption, the same model was used to predict the tissue concentrations of digoxin. So first, basically the PGP transporter is present in the kidney and liver tissue, and we had sufficient data to verify the findings, the tissue concentration simulation. So in both kidney and liver, the model well recovered or captured the kidney and the liver tissue concentrations, and also in case of other tissues, for example heart and muscle, the model was able to recover the tissue concentration. So you might be wondering that the PGP transporter was not incorporated in the heart or muscle, but it's still the overall tissue distribution, drug distribution is important to well capture the digoxin concentration in other tissues, even where the transporters are not involved. So once we have that confidence in the tissue concentrations in the elimination organs, we can also use the same model to predict the tissue concentrations in organs where the specific drug transporter is not involved. Let's come back to the very first question we asked about extrapolating the animal data to humans using PBPK modeling. So in case of rats, the rat PBPK model shown for digoxin, we used the abundance to inform the intrinsic clearance in the model. And the same equation, the Michaelis-Menten equation, can be used by providing this relative abundance factor to extrapolate the model to humans. So here in this case, the abundance data in human and the abundance data in rat is used to calculate the intrinsic clearance in the human, and the Km information, it can be assumed same if the active sites of the proteins are orthologous between rats and humans. However, if not, then we would like to have the information on the Km in both humans and rats. So using this equation, we can actually perform this extrapolation, which is more mechanistic in a sense that it's not merely based on the body surface area or body weight, but rather accounting for the interspecies differences in the protein abundance, the DMET protein abundance, especially when a drug is majorly metabolized or transported. So we also perform the analysis of the person sequence similarity of the DMET proteins in the rats compared to humans, and it was found that on the average 70% person sequence similarity was found for all the analyzed DMET proteins between rats and humans. In the current model also for digoxin, considering that the proteins are orthologous, same Km values were used of the PGP transporter and the CYP3A enzyme. So now let's review the key conclusions of this talk, and I hope I'm able to convince you today that the tissue drug concentration is crucial for evaluating drug efficacy and toxicity at the target tissue. But measuring this tissue drug concentration is often not feasible. So as a reliable alternative, a combination of in vitro assay, quantitative proteomics, and PBPK modeling can be used to predict tissue drug concentrations. As a proof of concept, a proteomics informed PBPK model was developed for digoxin in rats, demonstrating its ability to predict tissue drug concentrations. In drug development settings, this approach can be applied by obtaining DMET in vitro and proteomics data for the drug candidate, then integrating this data into a PBPK model, and finally using this model to predict tissue drug concentrations at the target site, thereby helping in guiding the dose optimization and potential clinical trial outcomes. So where are we leading from here? It's now a changing era of PKPD to PBPK QSP modeling. So mostly the PKPD analysis is based on systemic drug concentration, but using the PBPK QSP modeling can enable PKPD based on the tissue drug concentrations. It can also help to understand the mechanism of drug action at the tissue level, which can then help to establish confidence in target occupancy profiles as well. It's a famous the quote from this paper in Nature Medicine. The mechanism matters, and I'm just going to read the highlighted line here that the path of drug development is accompanied with different hurdles. Gaining a clear understanding of how a drug works before it enters clinical trials is the intelligent route to drug discovery, and that could increase the likelihood for drug success. So hopefully by integrating the PPK QSP modeling in order to predict the tissue concentration and using tissue concentration as a driver for pharmacodynamic response can help us increase our confidence in the possible mechanism and therefore increasing the likelihood for drug success. Thank you very much for joining this presentation again, and I'm happy to take any questions.
Thank you, Sheena. And we do have a few questions. So let me start with some of the ones that came up early in the talk. You mentioned that you did proteomics for liver and intestine. Did you look at other tissues such as brain or kidney and do the proteomics for those as well? You're on mute. Sorry.
Can you hear me now, Rebecca?
Yes.
Okay,
Yes.
thank you. Yeah, no, thanks for the question. And I wanted to share that the main tissues that we wanted to focus was liver and intestine because of the fact that these are like also like the major elimination organs. For kidneys, we do have data, but it was generated for some selected proteins, so it was not included in the publication itself. There are certain publications from the lab, like from the Washington State universities, and also other researchers have published data in different organs as well as different preclinical species as well. But for our focus was mainly the liver and we wanted to highlight basically the regional intestinal segments. So that was our like the key highlight of the paper.
The other part of this question that came up was, are there plans for other species?
Yeah, no, I just like I just shared that as a path forward, certainly a comprehensive analysis of all these tissues is certainly needed and needs to be performed. So this is certainly a good future direction to include more preclinical species and also other organs responsible in drug elimination.
Um, and there are a number of questions that have come up that are very similar, and they ask, what about differences between in vitro and then some of the cell culture lines that are often used for these proteomics?
Good question. Yeah. No, so basically the differences between the in vitro system and the in vivo system, and that's where the in vitro to in vivo extrapolation is needed. And to do the same, proteomics is again is needed to basically provide those scalars. So depending on the in vitro model used, there can be different scalars. For example, I talked about ref, which is the relative expression factor, and it's a scalar based on the protein abundance basically we found in the tissue versus the in vitro protein abundance. And these scaling approaches are beneficial in cases when there are differences in the protein abundance basically. So to account for that the protein abundance between these systems, it's important to provide these scalars. And there are other scalars including relative activity factor, which is based on the differences in the activity, functional activity, or basically the clearance. So these scalars are important there, and proteomics information is certainly warranted to inform those scaling factors.
And there is a question, have you verified the tissue concentration predictions with your approach, since there's been a lot of rat tissue concentrations published, I gather, for some of these drugs?
Okay, so just to make sure, so it's based on uh, the digoxin model that we performed. It's more or less the proof of concept of the data that was generated in rat tissues, both the liver, intestine, and the in-house, the kidney data for the PGP abundance. But certainly the future direction, again, is basically to verify for different proteins that we have identified in our global proteomics method. So uh, other drugs which are transported specifically by different drug metabolizing enzymes and transporters can be used to verify these protein abundance, and further the PBPK modeling and can be used to basically verify those tissue concentrations wherever we have the literature reported data.
Do you think we could use available proteomics databases for these measures, so that we wouldn't have to do the proteomics every time we changed among
Certainly.
rats?
Yeah, no, that's a good point. I mean, there have been some literature data already, and including the one that we have published in Molecular Pharmaceutics. So these literature reported proteomics data can certainly be used to inform these proteomics informed data, like the proteomics informed PBPK modeling, and that's the purpose of having this comprehensive exercise, I guess, to basically provide that information in the literature. And certainly this needs to be leveraged in the in performing these tissue drug concentration prediction.
For the transporter kinetic assay measured using transferred cell lines, would you correct the abundance measured in that system with the plasma protein content if the transporter locates on the membrane?
Yep. No, that's a good point. So I mean, so like the protein abundance that was done in the homogenate versus the membrane protein fraction. So that's where, I mean, the how the methodology is there for the protein quantification plays a role. And in our paper also we highlighted that with with the targeted proteomics, we performed the proteomics experiment on both the homogenate as well as the membrane protein fractions. So if we have that information, the proteomics information in the membrane protein fractions, that can be used in the modeling. And again, the use of the ref scalars like the relative expression factor can be used to basically normalize the abundance and correct those differences in vitro versus the in vivo protein content.
Do you consider the variability of the targeted proteomics, i.e. the error in the measurements for the PBPK model?
That's a good point, and I think the nature of the PKSIM platform that we have, it's based on the relative abundance like method. So the abundance that was incorporated was basically as a relative factor of the lever, but within that population, for example, the rat population, there is an inherent variability, or the basically the percent CV coefficient variation across all the physiological factors that has been considered in the modeling exercise.
Could you please highlight the differences between the bottom-up approach and the top-down approach? Which did you adopt?
Good question. So as one of the slide illustrated, it basically, the bottom-up approach is mostly starting from the in vitro experimental data to get the in vivo predictions, and this is not only applied for the clearance, but rather it can be anything like even the intrinsic solubility. We can start from the in vitro data, use the modeling tools to get the physiological relevant or the in vivo parameter. So that's basically the bottom-up approach. If we are talking about the top-down approach, then we start with the in vivo, the parameter that is observed in vivo to calculate the in vitro parameter. So in our modeling exercise, we basically use the bottom-up approach in a sense that we use the proteomics information that was generated in vivo to inform the drug disposition, and also the middle out, basically in a sense that we are also getting these the intertissue abundance incorporated in the model. So basically, by using these relative abundance values, and also like the proteomics information that was generated as part of the modeling exercise we did, we try to do the bottom up proteomics, and that's the essence of doing the PBPK modeling. So it's, if it is bottom up, it basically it's more mechanistic as well. But in certain cases like the population PK analysis, sometimes we start with the in vivo observations, and that's also good in giving us the confident, and also to account for the variability at all the levels.
How does the clearance extrapolation strategy compare with measuring the clearance using human hepatocytes?
So the question is the clearance estimation approaches, basically.
Yes.
So basically, we use the in case of this digoxin model, we use the literature-based Km and Vmax values, and like the model was also accounting for the differences in the PGP abundance and also the CYP3 enzyme abundance, and also their differences within the different tissues, like within the intestine, how much PGP is there in the proximal segments versus the distal segments, and also in the liver as well as the kidney. So, but we started with the literature reported Km and the Vmax. So basically, it's basically to account for even the nonlinearity in the modeling system.
In your model, have you assumed the parameters or estimated or used ones from experiments?
Right. So basically, we try to use the literature observed data. So like in the case of intrinsic clearance, in order to basically get those, it was mostly the KM was all observed, but in certain cases the Vmax was corrected, It was corrected by the protein abundance in the PKSim software. It's an inbuilt the equations that I was showing in one of the slides, and also the basically we had the in vivo data, the observed data. So basically, the further modeling was done to match the observed data as well.
There are a couple of questions on which software did you use?
Yeah. Okay. So for the modeling purpose, it was the
Yeah.
PKSim software. It's an open access software for performing the PBPK modeling simulations.
And one audience member asked, did you quantify CYP1A1 in the liver and intestine?
I
I guess
I
they're
I
interested in the specific.
Okay, so I mean very specific, and it's been a time since I last saw the actual data. But so most of the CYP and enzymes, CYP and UGT enzymes, and as well as the phase 2 enzymes, they were basically scanned using the global proteomics data, and we found mostly the CYP enzymes and the UGT enzymes in the liver tissue, the rat liver tissue, as compared to those were higher in the liver tissue, rat liver, as compared to the intestine, which is similar to what we observe in the human liver as well, like the human liver having higher CYP content as compared to the intestine. But with just specifically the CYP1A1, I don't remember exactly, but I can check it out and maybe can have a follow up later also.
Was the proteomics data that you measured entered into a database so that it could be reused by the public?
Yes, that's true. Like it's already there as an open access, and so it was part of the requirement of the basically the publication also to provide it and upload it to the open access, basically the repository, and it's embedded within the publication also, like to get the proper ID number to access that open, like the open access proteomics repository. So that information is there in the publication as well.
And just for our audience, that publication will be linked to Sheena's webinar on the webinar archive page, so that if you want to find it, it will be there. We have just a couple more questions about the polymorphisms and how do you handle polymorphisms with this approach?
So definitely, I mean, there are publications, nice publications from different institutes, that the genetic polymorphism can be handled pretty much the same case as we did in the case example of digoxin. So basically, the differences in the protein abundance can be provided in the modeling software. So for example, in case of the rosuvastatin PBPK PD model, like in different OATP1B1 genetic polymorphisms associated with OATP1B1 transporter, and due to that genetic polymorphism, there were like the poor, intermediate, and extensive transport associated, and basically it was shown that the proteomic levels were different for the OATP1B1 wild type versus the heterozygous or the homozygous deficient. So that information was provided in the software, and the software being used was SimZip in that case. And most of these the platforms that we have, they have these information embedded also, like what kind of changes do we expect in different genetic polymorphic subjects. So that information is sometimes embedded in the modeling tool that you are using, or if not, then even the data that is generated in-house with the proper, matching the units and everything, we can use that data, the proteomics data in the model as well, to basically correct for those protein abundance differences across the genetic polymorphic the subjects.
You did this proteomics in healthy animals. Would you expect it to change given a disease state?
That's a good question, and the answer is the short answer is yes. The protein abundance will definitely differ if we are comparing healthy versus disease population, and again, there are and many different publications studying the differences in the protein abundance across the liver impairment, renal impairment, and in different spatial populations as well. So the protein abundance will change. Other than that, other like the physiology will also be different in disease population as compared to the healthy volunteer. So for example, the protein binding, blood flow, and basically the shunting mechanisms, they will also differ. So that also needs to be accounted in the PBPK software.
So going forward, are there plans to expand this type of research and enter the information for public use?
Yeah, no, definitely. So I guess that's much needed. There are a lot of gray areas is still. I mean, there are different specific population and also disease conditions that needs to be investigated in more depth, especially the differences in the protein abundance needs to be well characterized. There has been a lot of research already, there. The data is there, but we might need to consider, for example, the sex effect as well. So having those understanding how between males or females, how the protein abundance can be different, and also with respect other than the disease population and the like the healthy versus the disease, as well as the sex effect, other DMET proteins, for example, especially the transporters, they needs to be well characterized across all the different tissues, uh, within the tissues in the same individual, and also in different subsets of population. Definitely that can help in designing the trials, and even if we have sufficient data, verified literature, then we can the next step is to using the PBPK model to support the clinical trial decisions, and even waiving the clinical trials.
And I think we have time for just one more question. This is somewhat technical. They ask how many replicates or individual animals were used to obtain the abundance data profiles, and do you think that adding additional replicates would lower the variability, or is this an assay issue?
That's a good point. Good point. So I mean, the replicates were the experiments were done in triplicates, and so we had the male Sprague-Dowley rats. But so it was like 6 animals, I guess. In that 5 or 6 animals, and the experiments were done in triplicates. But definitely, if we have more, the sample size is more, and even having that the female rats included, that will also be more a better information, and this is something which can be further as a future direction can be performed as well.
Thank you, Sheena. And just to let the audience know, we will pass the questions on to Sheena, so there can be some follow-up, particularly for some of the more technical questions. I'd like to thank you, Sheena, for a wonderful webinar. We really enjoyed having you here, and we hope that you and all of our audience members will come back for a future webinar. Thank you so much.
Thank you all for attending the talk and for very nice and thought-provoking questions.
Have a great day, and we'll see you again soon, I hope. Thanks, everyone.